Good morning, students. I hope that you all are doing good and please stay safe during this COVID time and follow the guidelines given by the government and if necessary wear double mask. So today we are going to start a new chapter and the chapter is Transpiration. We are doing plant physiology and in the last chapter we have done about the absorption by the roots, in which we have seen that how water and minerals are transported from root to different body parts of the plant. Now this term transpiration is not new for us because we have already done this term in the previous chapter, but this chapter is totally related to the process and this process is the transpiration. Now first of all, why the transpiration is useful for the plants? As you know that there is some pressure which is exerted by the root so that the water and mineral can go upward. Apart from this, from the top of the plant or the top of the tree, a force is also enacted on the water and minerals, and that force is known as the suction force, due to which the water and minerals they can move to certain point in the part of the plant. That means they can reach to the tip of the plant, and this is very important for the translocation of water and minerals in the plant. Apart from this, the transpiration is also very helpful, as it is helpful in the cooling process, because as you know that the water from the plants is getting transpired by the leaves of the plant mostly, due to which the temperature in the plant becomes stable and it will make the cooling sense. Just like from our body, when the water gets evaporated in the form of sweat, we feel cool. In the same way, the plants, they are also getting a cooling effect, and which is helpful in survival of these plants on the earth. So transpiration, as you already know, that basically a phenomena by which the water get lost from the body of the plant in the form of vapor, and basically this occurs from the tip region. So according to definition, process of loss of water in the form of water vapor from the leaves and the aerial part of the plant. So you have to just learn this diagram, which is very important. Now here a demonstration of transpiration is given and you can learn the whole paragraph. However, I am giving the main summary of this experiment. See here, a setup is given in which a plant is placed inside a pot, and this plant is covered with a polythene bag. Now why the plant is covered with the polythene bag? Because when we will keep this plant outside, so the plant will start transpiring, and since when the plant is starting transpiring, you will find that the water get changes into vapor, and when this water vapor get condensed in this plastic bag, you will find that tiny droplets are formed on the surface of this polythene bag. So that's why the plant was covered, so that we can see that how much the water is transpired by the plant. And as you know that the suction force will be there and from the tip of the plant. So the tip of the plant, basically the aerial part of the plant, will start losing the water in the form of vapor, and when this vapor it get condensed in this polythene bag, you will find that it get changes into tiny droplets of water. So this is basically the experiment, or we can say the demonstration of transpiration. However, if we want to take a control of this experiment, in the experiment we can take only the polythene bag but not the plant. If there is no plant, that means there will be no transpiration, there will be no air bubbles found on the surface of the polythene bag if the source is missing. Source means if the plant is missing, in other experiment the polythene bag will not show any drop of water. So this is basically the demonstration of transpiration. And here one more thing is given. Here it is mentioned if it tests by dry cobalt chloride paper. Now what is the role of this cobalt chloride paper? It is basically, it acts as an indicator of the presence of moisture. So this term is very important to learn, cobalt chloride paper, and many times in the exam it is asked what is cobalt chloride paper. It acts as an indicator of the moisture. So here you can see that cobalt chloride paper is an indicator of the moisture, and it is basically blue in nature when it is dry, but as soon as the moisture content present on any surface, it will start changing its color from blue to pink. So if the cobalt chloride paper get changes into pink, that means there is a presence of moisture. Now here another experiment is set up for the process of transpiration, and also you can read the whole paragraph. I am just giving the main summary. You can see that figure 5.2a1, in this the potted plant is there and potted plant is placed inside a jar. Now as the temperature goes higher, the water will start changes into form of vapor, and from the aerial part of the leaf the vapor will go into the jar and it get condensed on the surface of the jar, and you will find the tiny droplets in the experiment A. In this experiment you will find the tiny droplets of water on the surface of the jar. Similarly, the experiment was same, but here apart from this they have also used a dry cobalt chloride paper. Now since the water is formed, the water droplet is formed because of the transpiration, after some time this cobalt chloride paper will become pink in nature. Earlier this cobalt chloride paper will be blue in nature, but after, when it exposed to the moisture, the cobalt chloride paper will turn into pink color. So that means here in figure A and figure B, in both the process of transpiration is taking place. And how we can say that? Because of the result which we are getting. First, the water droplet is finding on the surface of the jar. Secondly, the cobalt chloride paper is turning from blue to pink. Now in the third one is the control. And why the control is always taken when we are doing any experiment, so that we can compare our result. Now in this, the blue cobalt chloride paper is there, the jar is there, but since the source is missing, that means the plant is not there. और सिर्फ जबकि यहाँ पे कोई plant है नहीं, so फिर यहाँ पे transpiration नहीं हो सकता, because the source is missing. There is no plant from where the water can get evaporated. So if the source is missing, ultimately this cobalt chloride paper will be blue only, and there will be no appearance of droplets on the surface of the jar. So this is basically the experiment. If you will read this whole experiment, you will be able to understand this. Now what is basically the proof which we are getting that yes, the transpiration is taking place? See, by taking the 3 well jars as described above, there is double visual proof of transpiration. What are these double visual proof? First of all, the condensation of the water vapor into droplets, कि जो हमको पानी की droplet दिखी थी on the surface of the jar. And second, the change in color of the cobalt chloride paper. That means earlier the paper was bluish in nature, and when it gets moisture, it will start changing its color and become pinkish in nature. Now next is a very important topic of this chapter. This is measurement of transpiration. How we can measure transpiration? The first method is the weighing method. Now in this weighing method, a lightweight plant is potted before and after, and weighed before and after the end of a certain period of time. कि हमने क्या है, एक छोटा सा पौधा लेना है, and that plant we need to weigh in the beginning of the experiment and after the end of the experiment. And the surface of that plant will be get covered. That means the soil is get covered by the polythene bag, so that there should be no loss of water in the form of vapor from the soil. कि soil में से water vapor की form में ना change हो जाए, वो evaporation ना हो जाए, so हम उस surface को क्या कर देते हैं, cover कर देते हैं. Now after some time, but you will find that the weight of the plant which was kept for some time, like for 3 hours, 4 hours, you will find that the weight of the plant will start decreasing. Why is it so? Because after some time the plant will start transpiration, and due to which the leaf of the plant, they will open their stomata, and from that stomata the water will get changes into vapor, and it will ultimately result at the loss of the weight of the plant. So this is basically the experiment, the weighing method. Now this experiment is modified, and here you can see that in this jar is there, and this jar is fully filled with the water, and a plant is there, and from here the water is inserted into this jar. Now what you will find that after some time, the water which was initially at this level, the level of the water will goes on decreasing. That means where this water is going. If this water is going into the jar, that means the jar should be full and there should be no change in the level of the water. But since the water level is going down in this measuring cylinder, that means the water is going somewhere, and it is proven that where this water will go, this water will go from roots, and from root to the stem part of the plant, and from the stem part of the plant, that from the leaf the water will get changes into vapor form, and this means that the evaporation is taking place from the surface of the leaf, and that evaporation which is taking place on the surface of the leaf is known as transpiration. So after some time you will find that there will be decrease in the level of water in this measuring cylinder. So this is the experiment to show that the transpiration takes place on the surface of the plant. Similarly, another weighing method is there. In this, the plant is placed inside a test tube and it is filled with the water, and oil is also placed inside this, and as you know that since the oil is lighter than the water, the oil will start floating on the surface. And why the oil is placed? So that the water should not get evaporated, because as you know the temperature will rise, the water will start evaporating. To avoid this water to get evaporated from the surface, the oil is placed. Now what you will find that after some time the level of the water will goes on decreases, because the transpiration will take place from the leaves of the plant, and ultimately this water will goes on decreasing, and the level of water will be somewhat like somewhere like lying like this. Maybe it can goes down till here, till here, till here. It can be this experiment can be continuous as long as the plant is placed inside the test tube. But the condition is that in this there is no root present in the plant. That means if no root present in the plant, that means the absorption of the water will be less, because as you know that in the previous chapter we have done that for maximum absorption of the water in the mineral, the root surface should be enormous, and since here the absence of the root is there, so the process of the transpiration will be very much low. Now another very useful method for measuring the rate of the transpiration is the potometer, and this is one of the important topic, and so many times it has been asked in the board exam. The potometer basically it is a device which measures the rate of water intake. Remember this, it is used to measure how much amount of water is taken by the plant, and it is assumed that this water will be always equal to the वाटर लॉस थ्रू द ट्रांसपिरेशन, कि जितना पानी इसने प्लांट ने एब्जॉर्ब किया है उतना ही पानी जो है वेपर बन के प्लांट से बाहर चले गया है. नाउ देयर आर वेरियस टाइप ऑफ पोटोमीटर, बेसिकली फार्मर्स पोटोमीटर, गैनोंग पोटोमीटर, डार्विन पोटोमीटर एंड गैरोस पोटोमीटर, बट वी यूजुअली रीड अबाउट द गैनोंग पोटोमीटर. सो a setup of the Gannong potometer is given, and here you can see that a twig of the plant is basically placed inside this graduated measuring cylinder. Now the entire apparatus is all filled with the water, and a scale is there to take the reading. Now what happened in this experiment, here a air bubble is need to introduce. And how we can introduce? We will just lift this part or this part, so that due to the pressure from the air into the water, air bubble will form, and the air bubble will go into this measuring cylinder, and from here we can take down our reading. So how we can introduce an air bubble? This is done by lifting the bent capillary tube above the colored water. So that means this capillary tube, it need to be lifted above the colored water, that means this water, and due to which the pressure from the air will go into this measuring cylinder, and as a result a air bubble will introduced in this measuring cylinder. Now after some time you will find that the air bubble, it will start moving from this region to this region, and finally it will reach to this region. What it shows? It shows that the water which is going inside in this measuring cylinder, it always equal to the amount of water which is released by the process of transpiration by this twig of plant. That means here the process of the transpiration is continued, and the water is going from leaves into the environment by the process of transpiration. That means it is getting converted from water into water from water to the water vapor, and here the water is coming from this jar into the capillary tube. So here one line you have to remember, that the reading on the capillary tube would given the volume of the water lost in a given time. That means the amount of water which is lost by the leaves is always equal to the amount of water which is absorbed by this or intake by this capillary tube. And what is the use of this reservoir? That after reaching a certain limit, from here the water it gets released, and due to which the air bubble which was introduced into this capillary tube, it comes back into its normal position. So this is the role of the reservoir. So basically the photometer is used to measure the amount of water intake by the plant. Now here one important thing is given, and please remember this: photometer do not measure the water lost during transpiration. So it is not given that how much amount of water is lost, but it is measuring amount of water intake by the leaves. Now one thing you should know, that this diagram is basically asked in the board exam, so you must know each and every part of this diagram, and you must know the functions of each and every part. Now what are the precautions in the use of the photometer? Now there is some precaution that is necessary while doing the experiment. First of all, the photometer should be made watertight. There should be no loss of water from any region of the photometer. That means the cylindrical tube or the meter scale, it should be properly fixed, and there should not be any leakage. Second, the twig should be cut obliquely. So the twig, that means this plant part, it should be cut obliquely. And why is it so? So that the largest surface area of the twig will be there to absorb the maximum water intake, and underwater to avoid suction of the air bubble into the twig, which will stop the absorption of the water into the xylem. And the second thing to remember here, that the water bubble or the air bubble should not enter into this twig, because it will cause the hindrance, and since it will cause the hindrance, so from the phloem the water cannot be translocated further. Now what is the limitation? There is some limitation of this photometer, and first of all, introducing the air bubble is not very easy. That means it is not easy that you will just lift this part up and the air bubble will get introduced. No, because sometimes multiple air bubble can be introduced, sometimes the air bubble cannot be introduced properly. So this part is very much difficult in case of photometer. Second, the twig may not remain fully alive, because since here is no root present on the twig, so the twig cannot survive for a long period of time. That means this experiment should be done very fast, and any changes in the outside air temperature may affect the position of the air bubble in the capillary tube. That means if there is any change in the temperature, suppose the temperature goes down or the temperature increases, this can cause the change in position of the air bubble. So these three things are basically the limitation of the photometer. First thing, air bubble cannot be introduced properly easily. Second thing, the twig, it cannot alive for a long period of time. And third, If there is any change in the surrounding, it will affect your reading. That means the position of the air bubble in the capillary tube will get affected. So this is basically the Ganong photometer, and this part is generally asked in the board exam, and sometimes this question is generally asked. So students, I hope that you are able to understand today's topic. If not, no problem, we can discuss this part in the Zoom session. Till then, take care and have a great day. Thank you.